Do you know? Great, thanks everybody. We'd be happy to take questions from the audience to start off with, and then I guess I was going to start with a question about the lentiviral gene therapy, maybe for Dr. Duncan. With the timing of when cells are collected and then when they're transplanted, is there a window of time where you can test the cells to see if there's an insertion event before you use that clone or population of cells?
I think that's a great question. There are certainly samples that are saved, and what's challenging about it is that that's not, they've looked back and you don't identify those integrations there, and we don't know that they're all harmful. And so what we see is we follow integration site analysis prospectively in patients, and it's changing all of the time. And so we have some patients who, if you look at their integration site map, they have what looks like a fully clonal population and no evidence of disease at all. And then there are other patients who you watch over time, and that integration site changes and grows steadily. So I don't think that there's enough data for us to understand what that would mean, and it's not even necessarily present at that moment in a level that you can find. When we were doing the clinical trial, it took about 4 to 5 weeks to get the cells back. Now, after FDA approval, because of differences in release criteria and testing for the product, it takes about 8 to 10 weeks, so much longer, which has led to a whole other conversation, just like the financial piece of it, sort of that manufacturing. So I think we just still don't know enough how to change or decrease that risk. It's a great question, though.
Sounds like a mess. Other questions?
Hi. It's to follow up on Dr. Fatemi presentation, and it's for all the clinicians here. I'm working for the state lab on the lab side. Is it still good to screen for female for XALD? I know that in Europe you have, is it the Netherlands who screen only for male? Just a thought.
Not me.
Start out.
Okay. So this is a very good question. Whether, why are women or females being screened in the first place? And, in Netherlands, they decided not to. My personal view, I can give you, because I think this gets personal here, right? For somebody who hasn't done anything else other than ALD and doesn't know about anything else, and is not a geneticist, and does not think that we, in my opinion, be providing informed consent and opinions to people. We don't just make life decisions for them. But my view is that each individual female that is identified in the family almost certainly will result in the identification of several other males at risk that we can help. So from a public health perspective, it is exactly what it's doing. It's doing exactly what it's supposed to be doing. It's helping, identifying newborn screening cases. It's just a sort of an indirect, or she's sort of the collateral or the victim one could think of. Having said that, I think there is a major challenge, and I totally recognize that's not what newborn screening is for, but as somebody who sees these women in the clinic, there is not a single one who comes to us with oh yeah, I have ALD, doctor, and I knew that. They come in after a 10-year, 15-year mystery diagnosis. They are called chronic fatigue syndrome, POTS, dysautonomia, multiple sclerosis, crazy, psychosis, whatever you want to do, or MS is many of them have actually gotten immunotherapy. So I think it's really a problem that women with ALD are not recognized early enough. Of course, not in the newborn stage, but so I see these two reasons why I feel like it's justified, but it's a very, very ethical question.
Yeah, I would tend to agree. I mean, our data suggests that there's, I think it was 2.5 to 3 boys were picked up that were at risk for, some poor outcome with ALD from virtually every patient that was picked up. So, I mean, if you're trying to prevent the morbidity and mortality associated with ALD in the boys, even if you pick up a sister or a cousin or whoever, it gives you an opportunity to do those types of things. I mean, it gets kind of sticky in terms of if you pick up a girl that's carrier isn't the correct term here. So a young woman with with ALD, when do you pass that information along to them? And so those discussions with the families end up being kind of sticky too. They aren't going to have a phenotype, right? But if they're getting to the point where they're starting to have families and, you know, obviously those things end up being important as well. So it's kind of messy, but I would tend to agree that I would support screening the women as well, or the girls.
This is Jen in Chicago. My genetic counselor wants me to remind everyone that there was recently changes to the secondary findings on whole exome sequences reported. So when families opt in to receive these secondary findings, X-linked ALD and CTX were just added to that. So we are also going to find pre-symptomatic individuals that way. So, and it's good for that to be on our radar and for us to know that, because we are going to get more of these referrals when we find more of these pre-symptomatic or cases.
May Baker from Wisconsin. So we only screening for XLD a little bit shy away 2 years. I have like 2 or 3 instances is when we identified female and 2 or 3 instances the family said, we have a female in our family. We don't want to do anything. So in that situation, actually we accepting that because the family, they already know they have this family and adults. They're trying said, I don't want to do more. Is that okay?
No, go ahead. Sorry, I just want to make sure that we fully understand. So there, on that screening, you've identified a woman or a female who had a child who has.
No. Newborn screening identified the infant, the female infant, when we contact the family, and family said that we have multiple females in our family and we don't wish to do further. So in that situation, in general, newborn screening is you have to do confirmatory testing,
Yeah.
whatever. So in that situation, actually, we're accepting that. I just want to ask.
I think there's no right answer. I think it probably makes all of us very uncomfortable, and we would like to do that. But again, people have autonomy over themselves and their bodies and their families and know what to do. So I think the best probably we can do is educate and try and maybe revisit at different time points. But I think you can't force that piece of it, unfortunately. Or fortunately, you can't force things on medicine. You know what I meant?
Yeah, I think looking at the Dutch system, it's very expensive to actually do this because they measure the LPCs and they look for the X chromosome and count those, and then they decide we're not going to pursue any further for the ABCD1 sequencing. But another issue regarding the cost and the insurance. So there was a paper earlier this year. I think half of the authors were from Orchard, making the case that it is actually beneficial to society that you screen, and there's basically cost effectiveness in screening for and treating patients with MLD. Would that help at all with the insurance question? Those kind of studies?
Sorry, MLD or ALD? MLD. Well, yeah, who knows what's going to happen with the insurance side. So I think the major obstacle is the out-of-state Medicaid piece, because they reimburse poorly and we're not obligated to treat the patients. And so you have to jump through all these hoops to potentially clear a patient to come in. And so I don't know that the state would say, I mean, they may say, I don't want to pick on a particular state, but they may say that, well, we're going to reimburse you, but the hospital's going to say, well, that's insufficient, so we're not going to move forward. And so, I mean, that's kind of the catch-22 is you got to work out some methodology by which the state is going to do what they need to do to compensate the treating hospitals sufficiently to make it worth their while. Otherwise, the hospital's under no obligation to move forward and lose money. Is that kind of your situation?
Thank you, sorry. So we, it is the same thing that you have to do single case agreement, of course, to do all of those pieces. I think every hospital is going to look at it differently. I think our hospital looks at it as part of our mission, not necessarily for leukodystrophies, but across gene therapies, because that's a platform they're trying to build. But the hospitals also have to be willing to even handle a $3 million hold on that, because once you've gotten that product and you haven't gotten reimbursed yet, the hospital is kind of living, in that debt for a while. So I think for large hospitals, where there's an appetite, I think it's okay, but every state is a little bit different. And not again to call out a state, but the first patient we did commercial gene therapy from was from a state that reimburses very poorly, and we did have to argue and say they cannot get this therapy elsewhere, and had to write letters of necessity saying why an allogeneic transplant for that patient who did not have a donor, like all of those things. And I think that is something that hospitals and industry can work together to say, like, this is the packet that you need to say, and this is the appropriate literature. And sometimes it works and sometimes it doesn't. Even with commercial insurance, not all of them pay well because everyone's, Blue Cross has a different piece. So I know that hospitals are working on this and trying to get it together, but it takes a full team of people to do that. I think with approval for sickle cell and thalassemia, which are much larger populations, it's making some of this move forward more rapidly because it's getting more attention, although those are not as expensive.
Yeah, so the issue is always our broken healthcare system, where everyone looks at their own bucket of money and doesn't look at the overall healthcare cost and savings over time. Because I mean, if these patients die slowly with needing constant care versus maybe living a fairly normal life and can actually work and pay taxes, that might pay for itself. But I'm just dreaming.
No, I think you're right, but I think that having those papers they're never harmful. Like, so you could put that in part of that packet that goes to the, to the insurance companies and try. And some of them are better than others, of course.
I would just say some of the insurance companies, I think, feel like, well, so there's a lot of people that change their insurance every 3 to 5 years or something. And it's like, why do we swallow a $2.5 million cell product so that the next company won't incur losses? Do you know what I mean? So there's some of that kind of stuff, I think, that happens too, which is obviously unfortunate, but that's.
Well, presumably you win some, you lose some. Doesn't that average out across all the insurance companies?
Well, one would think.
You would think, except for it's pretty short-sighted for the, well, maybe that's insurance companies are short-sighted, but okay. Why does the Bluebird work for the transporter that doesn't?
I'm gonna let Ali tell you why that. He wants to know why. So is your question, why does bone marrow transplant or gene therapy work for ALD?
So cross correction. Elizabeth Neufeld cross correction lysosomal enzymes move from cell to cell, like from hematopoietic cells to cells that don't have the enzyme. But that's not true for the ABC transporter. So why does it work? So
now I've been trying to figure that out for a long time.
But presumably it's because the long chain fatty acids move from cell to cell.
Yeah, it has nothing to do with the very long.
How do you know?
It's clearly, what do you mean? It's clearly not
what's the evidence?
Cross correction sort of mechanism that you see with the lysosomal stuff, because that's, those are enzymes that,
for ALD.
Well, Ali's going to tell us in a second.
How do you know the fatty acid doesn't move from cell type to cell type?
Well, I mean, the fatty acids in the blood go down a bit after transplant. They don't go down to normal.
And maybe for MLD, it's the same thing. Maybe the sulfatides move from cell to cell, and it's not the enzyme.
I don't know. There's some protective mechanism by which having some donor-derived cells we think are the oligodendrocyte, is where the action is. But I don't know. I mean, biologically, it's totally different because the ABCD1 gene product is a peroxisomal membrane bound.
No, I get it. So
so it's not freely accessible through a mannose-6-phosphate pathway.
I understand. That's my question. Why does it work?
Well, I don't know. I was just saying that
there's only
it's clearly different, but I'll let Ali tell us.
What?
So I think there's a very good working hypothesis which is based on publication from Florian Eichler in 2005, where he showed that when you look at the microglial population in the brain of ALD patients with cerebral ALD, you see that there is highly activated microglia and normal appearing white matter. And then just at the edge of the lesion where we see this blood-brain barrier breakdown on the MRI, and we know it's a blood-brain barrier breakdown because we see that gadolinium gets into the brain. When you look at that edge, you see that there's microglial apoptosis. You also see that these microglia are sort of fully loaded with lipid particles. And then we also know from in vitro studies that we did years later that cerebral ALD macrophages and monophagocytic cells in general are highly reactive. When you stimulate them with very long-chain fatty acids, they have about a 20-fold increase in TNF-alpha as opposed to control cells, high IL-6 response. So they're pro-inflammatory cells in ALD due to the gene mutation. The exact mechanism why they're in that pro-inflammatory cells that then undergoes apoptosis, we don't really know. But the idea here is that you're replacing that monophagocytic population in ALD with doing the transplant. And then there's also a secondary theory, which is a little weaker, in my opinion, where there's also an endothelial cell dysfunction. So Patty Mussolino from MGH has shown that if you silence ABCD1 in endothelial cells, they become very leaky. And so they downregulate all their connexins and so on. And so that may explain the blood-brain barrier disruption. And there is, I guess, some data that hematopoietic stem cells may be able to become also endothelial cells in addition to migrating into the brain, become microglia. So it's sort of that we don't ever see a so from all the autopsies that are available, I'm not aware of any reduction of very long-chain fatty acids in the central nervous system following transplant. So that would imply that there is likely no sort of shifting in very long-chain fatty acid metabolism globally. So it's more like a targeted cellular response. It's really a microgliopathy. That's my opinion.
Okay.
Yeah, and I would just kind of mention that we're hampered because none of the models work, right? So the ALD mouse models don't develop neurologic disease, and I've heard there's a couple of rat models that didn't either. So we don't have a good way of looking at the biology of it.
Paul, I have a question for you since you mentioned the cost of the MLD treatment. How did they come up with $4.25 million as the treatment? It just seems kind of arbitrary. And then a follow-up to whatever your answer is would be, do they understand that they're biting the hand that feeds them?
What was the last part? Sorry.
Do you think the company understands they're biting the hand that feeds them?
Well, some of the Orchard Therapeutics guys are here, but you know, it's tough, and I don't want to, you know, I'm not part of the industry side of things necessarily, but you know, I heard that the Bluebird trials for ALD cost like $400 million to run, and took 10 years to get licensed. And so, you know, you got to make up all that venture capital that was expended over a long period of time. Plus, you've only got a finite number of patients you're going to be able to treat anyway. So, and you know, part of it comes from if there's a patient that you would otherwise expect to go on and have progression of their disease and incur a ton of medical costs and whatnot, and you could prevent that, then that's on the positive side of things too. You know, if someone's going to have a long, healthy, productive life and pay taxes and do all that other sort of stuff, then, you know, that's on that side of the ledger as well. But I mean, it's tough, like we were kind of talking about before. I mean, the cost benefit ratios for doing these kinds of things is just really difficult.
Hi, thanks for the session. Jen Orthman from Penn in Philly. Just curious if you think that gene therapy for adults who develop cerebral ALD or MLD is feasible or a good idea?
Well, I tried to talk them into letting us do it a pilot, actually, because I think, you know, Ali was saying that, you know, I'm a kind of proponent. You know, the patients with ALD that are adults, a lot of them have AMN and they may be in wheelchairs or whatever. The toxicity of transplant in a population like that is very high. And if we could use something that's less, you know, toxic and get them through I mean, the biology presumptively is the same. I mean, Eric was kind of saying before, it looked like that as you get older, your disease may progress a little less quickly too. So maybe I have more opportunity to stop things. It would make sense to be able to offer the gene therapy for the ALD patients, but I don't know if we'll have the opportunity to do that or not.
No, I mean, there was a lot of discussion.
Yeah.
We all wanted it, but.
Yeah. And I think some of it too, it is really tricky, especially people not going off labels. We would certainly have to do it on study, but we had a patient who was 18 years old, 17 years old and approaching 18. And we even knew that was going to be a risk. So we had to rush as fast as we possibly could to get that in, at least that approval before like the birthday. So I agree if we could do that and certainly do it as a pilot. But it's tough right now, I think. I think also the funding and such for trials in rare disease 10 years ago with gene therapy, I think there was people were thinking about rare disease as a way to prove your concept of what you can do now. And there's been this huge, with your product, with your gene editing or insertion, and there's been this big shift away in recent years from rare disease towards things like sickle cell. All of these diseases need therapies, but it has really been a really palpable change from where the focus lies.
Yeah, I just wanted to jump in on the cost of gene therapy. I think the challenge is it's anchored by the cost of a life that is affected by the symptomatic disease. And so that cost for MLD, which is the highest drug ever, is anchored into an evidence review by ICER on the cost of having a life with affected MLD. And so it's not a cost recovery price, it's a benefit gained from the therapy price, which I think causes this just like increasingly exponential cost of therapies. So, I mean, there was a formal review. It was really a bizarre process. They're trying to, in Europe, they anchor approvals to cost and it's integrated into the process because they have the nationalized or the universal healthcare. For us, it's completely independent, right? Like we approve and then we cost set as an independent, less transparent process. So ICER was built to try and fill that gap in the United States, where they have economists and academics and families come in and weigh in on what the burden of having the disease is, and they set a price point. So at least it's an attempt to make it better, but I think they anchored to symptomatic kids, not to the cost of development.
Me? So I'm gonna one-up Paul Levy's question and cause a lot of trouble here. So bile acid's pretty cheap, deoxycholic acid, it's dirt cheap, it's about $1 per gram. So the cost to treat a CTX patient over their lifetime will greatly exceed $4.5 million. That's an interesting thing to get your head around. And all you're doing is giving them deoxycholic acid, which is dirt cheap. So I have no idea where these prices come from. I just want to say that again, the cost over the lifetime of treating a CTX patient with something that's dirt cheap is going to exceed $4.5 million one-time cost for MLD. So yeah, the prices are absolutely insane and make no sense at all.
This is a CTX question. It's been kind of in my mind for quite a while now. The range of disease severity seems like, from infantile to adult with kind of a large number in adult.
We just barely hear you, so.
Oh, okay. Going back to the CTX therapy in newborns identified through screening, how will you know which babies to treat versus hold off treating versus how do you monitor when there's such a spectrum for disease?
So all I can say is refer back to the Delphi study, which was worldwide expert consensus study. And the idea is that the accumulation of cholestanol and bile alcohols is associated with the clinical signs and symptoms of disease, and those are elevated from birth onwards. So the recommendation was to treat from birth onward, and the risks of the medication are low. Don't know if that answers your question. So there is no waiting to see, and the monitoring was clinical monitoring over time, monitoring for biomarker response because it goes down, with the treatment when the treatment's effective, and liver monitoring, and that was the monitoring that was recommended in the Delphi study.
Okay, so there's really no predictive power to which variant or what form of disease people are going to have and what we're actually, I guess.
Yes. So currently there's been no
Preventing.
genotype phenotype correlations identified. There's been none of the biomarkers are prognostic for severity of disease or how that disease will progress. I mean, they're good diagnostic biomarkers, but this information is still to come. We don't know this yet.
Just one last thing. What do you think the compounding, issue will be with trying to get pharmacists to compound this drug for use in babies compared with adults?
So, as I mentioned in my talk, it is approved, it's indicated for pediatric use in Europe, both chenodeoxycholic acid and cholic acid. And, as we discussed before, there are physicians with a lot of, or not a lot, but relatively large experience treating infants with those medications. And I'm hoping to put them in touch with you for their experience. It was unfortunate that the CDCA by the FDA approval was only in adults, but at this point we waited so long for the FDA approval and it was such a challenging clinical trial that I'm very glad that there is at least the approval to treat CTX with CDCA, and hopefully the sponsor will follow up and apply for that pediatric indication as well. But again, I can only go back to the Delphi study and the expert consensus amongst experts. And again, CDCA has been used as the standard of care for the last 40 years since the clinical trial in the early 80s. It's safe and the expert consensus is treat from birth onwards. Sorry, Joe, I don't know if that answers your question.
A quick question for Dr. Fatimi. Right. Do you think that there is a role for liraglutide either after not during, probably during transplant, during conditioning or other things, but afterwards, particularly in patients who might have a higher or less score, or people were concerned about progression during that time, because we know it doesn't happen automatically and it takes time. So I could think just sort of in the leukemia world, we've now moved to a lot of post-transplant therapies, and this feels pretty straightforward, not toxic to a lot of drug interactions or other things, and it might help some of those patients who have a higher, less score, or more advanced disease stabilize or get through transplant.
So before the Nexus study was designed, there was a lot of conversations with the company about developing a clinical trial that would actually go for advanced cerebral ALD. With the idea that, there's all this population that we can't treat right now. What are we going to do with them? And the decision was made not to pursue that based on some data that I haven't seen personally, but was told that when it was tried in some advanced cases, it didn't seem to be working. And so I don't really know. But it's also looking at the population in the Nexus study that Eric has been working on. It seems like perhaps the earlier you go, the better you are in general. And in the AMN population, if you say that the sway amplitude does matter, which the FDA discounted, again, it was only a treatment effect was there when you looked at those with the shorter disease course with AMN. So I'm not sure if it's going to help for advanced cerebral ALD. The question that you have is whether it could help in conjunction sort of right after transplant in those with a high score. I mean, I think you need to look at it. I don't think we have any data to say one way or the other. But you could also argue that so far we don't really have proven evidence that it's actually effective, right? Otherwise it would be approved already. So we're still on a chase. Yeah.